I'm Damla, and today I will be talking about our paper on Nanopore Sequencing Technology and Tools for Genome Assembly. And here are the QR codes for our paper. We published in Briefings in Bioinformatics, but we also have the archive version if you are interested. So, nanopore sequencing is an emerging and a promising single molecule DNA technology, and it can produce long reads and it provides portability, so that's why it's promising. However, it suffers from high error rates, and because of that higher error rates, the tools for genome analysis have critical importance, both for overcoming this higher error rates and also enabling fast and real-time data analysis. So in this work, we analyze multiple steps and the associated tools for genome assembly pipeline using nanopore sequence data, and we performed our analysis in multiple dimensions: accuracy, performance, memory usage, and scalability, and we revealed new bottlenecks and trade-offs that different combinations of tools lead to, and we provided guidelines for both practitioners and tool developers. So let's start with the nanopore sequencing technology and the advantages of it and how we analyze nanopore sequence data. So nanopore sequencing technology, as I said, is an emerging and a promising technology, DNA sequencing technology. And the first nanopore sequencing device, Minion, was made commercially available by Oxford Nanopore Technologies in 2014, and it's an inexpensive device, and it can generate long reads, as I said. And these days, these long reads can reach millions. And it produces data in real time. Also, it's a pocket size and a portable device, which is just a USB-sized device. And this is the Minion. So how nanopores perform sequencing? So what we mean by nanopore, it's a nanoscale hole. And between the walls of this pore, an ionic current passes. So when the DNA strand passes through the pore, it interrupts the current, and when we measure the change in the current, we can identify different nucleobases, because each nucleobase, ACGT, have a different electrochemical structure. So they affect the change in current in different magnitudes. So by looking at this change, we can identify the bases in the DNA strand. So why nanopore sequencing is promising, and what are the advantages when we compare with the prior high throughput sequencing technologies? First of all, it does not require an amplification step before the sequencing process, and it does not require labeling of the DNA or nucleotides. And with the help of these two advantages, it allows sequencing of very long reads, as I said. Whereas the high throughput sequencing technologies were generating billions of short reads, but the advantage for these technologies were they were accurate reads. So as high throughput sequencing technologies, nanopore also provides low cost and high throughput, but also, as I said, it's highly portable. But it has one major drawback, which is high error rates, and these error rates can reach 15%. But high throughput sequencing technology's problem was they were producing massive amount of short reads, and these short reads poses challenges to the further analysis, because we have repetitive sequences in the genome, and a single short read cannot span the entire region, whereas the long read can span. And with the help of covering these repetitive sequences, we can end up with accurate and more complete assemblies. That's why we focus on the genome assembly pipeline in this work. So we have the current signal data from the nanopore sequencer, and then how we end up with constructing the assembly. So we first need to convert this raw signal data into the DNA reads, which has ACGTs. So this step is called basecalling, the initial step of the pipeline. And then by merging these long reads, even to way longer reads, we first need to find the overlaps between each pairs of these base-called DNA reads, and then with the help of this overlap information, we can merge and then construct the assembly. But if we are not satisfied with the quality, accuracy of the assembly, we can further apply optional steps, read mapping and polishing. So with read mapping, we map the DNA reads, base-called DNA reads, against the draft assembly, and then by using this mapping information, further polishing is applied. But these last two steps are optional. So our goal in this project, as I said, is comprehensively analyzing the multiple steps and the associated tools to find the advantages, disadvantages, and bottlenecks of different combinations of tools lead to. And we performed our analysis in accuracy, performance, memory usage, and scalability. And we revealed bottlenecks and trade-offs for different combinations of tools, and we provide guidelines for practitioners so that they can pick the appropriate tools for their goals, and also for tool developers so that they can make better design choices to improve the current tools or the future tools. So let's define our experimental methodology in this work. So we picked 3 systems. The first one, which has 40 cores and 128 gigabytes, is used for accuracy analysis. So we picked this system because it provides parallelism and performing the experiment. Whereas we picked the last 2 systems, Desktop and BigMam, to perform our scalability analysis, performance analysis. And the Desktop is it's corresponding to a typical desktop server, whereas BigMam has a very large memory and lots of cores, so they represent 2 extremes. And while performing our analysis, we set our accuracy metrics as average identity, coverage, number of mismatches, and number of indels. So average identity is typically the similarity between the assembly and the reference genome, so how similar they are. And coverage is what's the percentage of the reference genome we are covering with the assembly we constructed. Number of mismatches is number of single base differences between the reference genome and the constructed assembly, and number of indels are total number of insertions and deletions between the reference genome and the assembly. And for the performance metrics, we use wall clock time, peak memory usage, and parallel speed up. So let's start our analysis for the first step, Basecalling. And for the first step, we picked 5 tools: Metriker, NanoNet, Scrappy, NanoCall, and Deep Nano. So the first 3 tools are Oxford Nanopore Technology's own tools, and they perform Basecalling with recurrent neural networks, whereas last 2, NanoCall and Deep Nano, are independently developed by research groups. And NanoCall uses hidden Markov models, whereas Deep Nano is also using RNNs for Basecalling. So how we performed our, how we showed our accuracy is, we completed the first 3 steps, and then we compared the assembly for each different combination with the reference genome, and then we grouped the results by Basecalling tools and end up with 3 different pipelines. And let's show the accuracy results for these 3 pipelines. In the x-axis, you can see the tools, the Basecallers for each pipeline, labeled as PL, A, B, C. On the left axis, you can see the percentage values for identity and coverage, and on the right axis, you can see in kilobase pairs for number of mismatches and indels. So when we look at these bars for the identity and coverage results, when we look at first 3, Metriker, NanoNet, and Scrappy, we can see that they all have very similar trends in terms of identity and coverage for all of the evaluated scenarios. Whereas when we look at the last 2, NanoCall and Deep Nano, they cannot reach the other 3 Basecallers' accuracies, and they have lower identity and lower coverage. So when we draw the lines for mismatches and indels, And when we focus on the points for the Scrappy, you can see that they have a lower amount of mismatches and indels, which leads to the conclusion that Scrappy has the highest accuracy with the lowest number of mismatches. And when we compare the performance of these base callers, we could not include deep nano in this analysis because it does not support multi-threading, and we couldn't include Metriker because it's a cloud-based tool, and we don't have the source code for it. So that's why we focused on Nanocall, Nanonet, and Scrappy for the performance analysis. So when we in the x-axis for both of the plots, you can see the number of threads can reach maximum number of logical cores for each system. And the y-axis represents the wall clock time in seconds. So when we compare Nanocall with Nanonet and Nanocall with Scrappy, we show that RNN-based base callers, which are Nanonet and Scrappy, are faster than HMM-based base callers, Nanocall. And also another observation from these plots is when we compare the single-threaded performance between desktop and BigMem, we show that desktop is approximately 2x faster than BigMem. The reason is that the desktop has higher CPU frequency, and the larger memory or the larger last level cache capacity of BigMem cannot make up for higher CPU frequency, which indicates that all of these three tools, base callers, are computationally intensive, more than memory intensive. So when we compare the memory results for these three tools, we show that Scrappy and Nanocall have a linear increase when number of threads increase, whereas Nanonet has a constant memory usage for all the evaluated thread units. The reason is that in Scrappy and Nanocall, each thread is assigned a different set of reads to perform the base calling, which increases the memory usage when we increase the number of threads. Whereas in Nanonet, computation is divided between different number of parallel threads, so that it's always constant. Because even though we have, let's say, 80 threads, the number of reads processed have equal with the single-threaded version. That's why it's a constant memory usage. So in summary, we showed that the base caller choice has an important role for overcoming the high error rates. It's important because it ends up with different accuracy levels. And base calling with RNNs provides higher accuracy and higher speed than HMMs. And also the newest base caller of Oxford Nanopore, Scrappy, has the potential to overcome the homocolumnar base calling problem, which is the repetitive regions, base calling the repetitive regions, which is a challenge because the Nanopore device cannot predict the length of these repeats, but Scrappy is targeting that problem, and with the minimum number of mismatches and indels, it is promising to overcome this problem. So let's move to the second step, read overlap finding. We picked two tools, GraphMap and Minimap, for this step. So what's different between these two steps, between these two tools are GraphMap partitions the entire read data set into k-length substrings, k-mers, and detects the overlap and stores them in a hash table with the positions and uses k-mer similarity to find the overlaps. Whereas Minimap partitions the entire read data set into k-mers, but only stores the minimum representative set of these k-mers, which are called minimizers, and then uses minimizer similarity to detect the overlaps. So the reason of storing minimizers is reducing the memory requirement. That's why they do not store all of the k-mers. So we did the same. We followed the same approach for analyzing the accuracy of these tools. And we end up with 5 different pipelines, A to E. Each of them have different base caller and a same assembler. And this y-axis and x-axis are same with the base caller plots. So when we group them, And when we look at the trend, we can see that, and it applies for the mismatch and indel trends. The pipelines with GraphMap or Minimap end up with similar accuracy results. So which tool we picked for this step doesn't make any effect on the accuracy results. However, we cannot say the same for the performance results. So when we look at the right-hand side, the memory usage, and the left-hand side wall clock time, and one quick note, we could not perform GraphMap on desktop server because of its high memory usage. That's why we can only show the results in BigMem. So when we look at the right-hand side, we show that although the memory usage for both of these tools are constant, so depend on the hash table size but not on the number of threads, by storing minimizers, Minimap requires 6x less memory than GraphMap on average. And this ends up with a speed up also because Minimap shrinks the data set, and then by storing minimizers, and it performs much less computation when spanning this smaller hash table. So it ends up with 2.5x faster than GraphMap. So as we showed, Minimap, by storing minimizers, is not losing any accuracy, but providing lower memory usage and thus higher performance than GraphMap. So the assembly tools, we picked Cano and Miniasm, the third step to construct the assembly. And the Cano and Miniasm has difference in its interior pipeline. So Cano performs error correction before constructing the assembly, whereas Miniasm skips this step because it's computationally expensive. So Miniasm, by lowering computational cost, it's directly depending on the accuracy of the uncorrected reads, whereas Cano, with the help of error correction, performs finds the overlaps and constructs draft assembly with the corrected reads. So this ends up with Cano having higher accuracy than Miniasm. So for example, Cano on average has around 96% accuracy, whereas Miniasm provides only 84% identity. But it comes with a cost, and Cano is greatly slower than Miniasm, like 1000x slower than. So what we can show here is there's a trade-off between accuracy and performance when we are picking the assembly tool. So what we can suggest here is Miniasm can be potentially used for fast initial analysis, and then the optional steps can be further applied to improve the quality of the assembly and to produce higher quality assemblies. So the last step, the optional read mapping and polishing tools, because of time constraints, we could not give the details, but you can find the details in the paper. But what we can summarize here is further polishing can significantly increase the accuracy of the draft assemblies, and pipelines with Minimap as read mapper and Raycon as polisher can provide a significant speed up over the BWM and Nanopolish pipeline. And Minimap and Raycon also results with high quality consensus sequences. So what we can imply for future from this analysis is the choice of tool for base calling is really important for overcoming the bottleneck of the technology. And RNNs perform better than HMMs in terms of both accuracy and performance, so they are more promising. And as we according to our findings, we showed that parallelizing the tool can increase the memory usage, but we can divide the input into batches, or we can limit the memory usage of each thread, or we can divide the computation instead of the data set between parallel threads so that it can prevent large increases in memory usage. Also, it can provide performance benefits from parallelization, which is important. And the last thing is, since Nanopore sequencing is providing real-time analysis and it's portable, we can use them for in-field analysis, like outbreaks, Ebola, Zika outbreaks, where resources are limited. Because of that reason, laptops may become a popular platform for running the assembly pipeline. And we should keep in mind that laptops have greater memory constraints, lower computational power, and limited battery life. So it's important to take these into consideration while designing algorithms or accelerators. And to conclude, as our motivation I showed today, nanopore sequencing is promising and emerging and promising technology because of long reads and portability. And it's important to have faster tools, and also tools that can overcome the higher rates of the technology. And we analyze multiple steps and tools, as I showed, in multiple dimensions, and we reveal bottlenecks and trade-offs that are important for both users and tool developers. Thank you.
Thank you, Damla. We have time for one question, if someone wants. Actually, I have a short question.
Sure.
You presented us the entire pipeline and a lot of tools. What do you plan to focus on next?
So currently we are focusing on read mapping, but the main bottleneck of the full pipeline is Basecalling, because it's the most time-consuming part, and it's the real, it's a critical part to overcome the error rates. So Basecalling is the most important step of the pipeline.
Okay, thank you.
Okay. Thank you, Damla.